Hello everyone. Let's start Ncert reading. Today's chapter is Cell Structure and Functions. He is the scientist G.N. Ramachandran. In the field of protein structure, he was the founder of Madras School. So, the chapter name is Cell, the Unit of Life. When you look around, you see both living and non living things. You must have wondered and asked yourself, what is it that makes you organism living? Or what is that an inanimate thing does not have which a living thing has? The answer to this is the presence of the basic unit of life, the cell, in all living organisms. All organisms are composed of cells. Some are composed of a single cell and are called unicellular organisms, while others, like us, composed of many cells, are called multicellular organisms. So, what is a cell? Unicellular organisms are capable of independent existence and performing the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, the cell is the fundamental structural and functional unit of all living organisms. Three things: fundamental, structural, and functional. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. Cell theory. In 1838, Matthias Schliden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of the plant. At about the same time, Theodor Schwann in 1839, a German zoologist, please correct it, a German zoologist, studied different types of animal cells and reported that cells had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of all the plant cells. On the basis of this, Schwann proposed the hypothesis that the bodies of animals and plants are composed of cells and products of cells. Schleden and Schwann together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow, 1855, first explained that cells divided and new cells are formed from pre-existing cells, omnis cellula e cellula. He modified the hypothesis of Schleden and Schwann to give a cell theory a final shape. Cell theory, as understood today, is all living organisms are composed of cells and product of cells. All cells arise from pre-existing cells. An overview of cell. You have earlier observed cells in an onion peel and or human cheek cells under the microscope. Let us recollect their structure. The onion cell, which is typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane-bound structure called nucleus. This nucleus contains the chromosome, which in turn contain the genetic material DNA. Cells that have membrane-bound nuclei are called eukaryotic, whereas cells that lack a membrane-bound nucleus are called prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both plant and animal cells. Various chemical reactions occur in it to keep the cell in living state. Beside the nucleus, the eukaryotic cells have other membrane-bound distinct structures called organelles, like the endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane-bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplast in plants and mitochondria, and on rough endoplasmic reticulum. Animal cells contain another non-membrane bound organelle called centrosome, which help in self-division. Cell division. Cells differ greatly in size, shape, and activities. Refer Figure 8.1. Red blood cell, round and biconcave, diameter 7 micrometer. White blood cell, amoeboid. Columnar epithelial, long and narrow. Nerve cell, branched and long. A tracheid, elongated. Mesophyll cells, round and oval. For example, mycoplasma, the smallest cell, are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer in length. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Remember this. This is important. Human RBC 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function they perform. So let's start prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, pleuro, pneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells. Figure 8.2. Here is the PPLO, size 0.1 micrometer in size. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, rod-like, bacillus are rod-like, coccus, spherical, vibrio, comma-shaped, spirillum, spiral-shaped. The organization of prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shape. A bacterium. The prokaryotes have a cell wall surrounding the cell membrane, except in mycoplasma. Mycoplasma does not have cell wall. The fluid matrix filling the cell is the cytoplasma. There is no well-developed nucleus. The genetic material is basically naked, not enveloped by nuclear membrane. In addition to this genomic DNA, the single chromosome or circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmid confers certain unique phenotypic character to such bacteria, that is, this character is resistant to antibiotic. In higher class, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotic cells, except for ribosomes. Prokaryotes have something unique in the form of inclusions. A special as differentiated form of cell membrane called mesosome is the characteristic of prokaryotes. They are essentially infoldings of cell membrane. So, next, cell envelope and its modifications. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. Cell envelope consists of tightly bound three-layered structure: outermost glycocalyx, followed by cell wall, and then the plasma membrane. Although each layer of the envelope performs distinct functions, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of their differences in cell envelopes and the manner in which they respond to staining procedure developed by Gram. That is, those that take up Gram stain are called Gram positive, and the others that do not take up Gram stain are Gram negative bacteria. Glycocalyx differs in the composition and thickness among different bacteria. It could be loose sheath called slime layer in some, while in others it may be thick and tough called the capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacteria from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by the extension of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, lamellae. They help in cell wall formation, DNA replication, and distribution to daughter cells. They also help in respiration, secretion processes to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores which contain pigments. Bacterial cells may be motile and non-motile. If motile, they have thin filamentous extension from the cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook, basal body. The filament is the longest portion and extends from the cell surface to the outside. Beside flagella, pili and fimbriae are also surface structure of the bacteria but do not play role in motility. Pili are elongated tubular structure made up of special protein. These special proteins are called pilin protein. Pili has pilin protein. The fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissues. Next, ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made up of two subunits, 50S and 30S units, which when present together form 70S prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to single mRNA and form a chain called polyribosomes and polysome. The ribosome of a polysome translates the mRNA into protein. Inclusion bodies. Reserved material in prokaryotic cells are stored in cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in cytoplasm. Example: phosphate granules, cyanophycin granules, glycogen granules. Gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria.